Good afternoon, or good morning, or good evening, depending on wherever you may be. We are so glad you could join us. Welcome all to our Navigating Controlled Correspondences to Support Generic Drug Development webinar. My name is Corey Adair, and I am with the Small Business and Industry Assistance, or SBIA program. I will be your host for today's session. Our SBIA program operates within the Division of Drug Information, or DDI, which in turn resides within the FDA Center for Drug Evaluation and Research, or CDER's, Office of Communications, or OCOM. Before we begin, we encourage you to connect with us. For your convenience, we've included QR codes to access SBIA on LinkedIn, X, YouTube, and via email updates. Scan the QR code with the camera on your cell phone. This will provide you with a link to SBIA on the platform for the QR code that you scanned. It's great to stay connected with SBIA social media platforms to learn about upcoming webinars, conferences, and regulatory information, and also to find recordings of past webinars and conferences. Now, let's turn our attention to some logistics. First, here is the link for our one-stop shop for answers to your technical questions, for downloading files, accessing the survey link to obtain certificate of attendance, and so much more. That's the same page that you used this morning for your online access to the event, so please bookmark that page. If you have any technical questions about this webinar, please check there first and you will most likely find your answer. We invite you to submit your questions to us so that we have a vibrant and productive Q&A segment, which is so crucial to these events. This is a learning opportunity, and I encourage you to take full advantage of the expertise that we offer here. When you do submit your question, please identify that individual presenter by prefacing your question with the speaker's name as shown here, @Yan. Please keep in mind that while we may not get to your individual question, they are always very helpful as we plan for future events. The final survey is the mechanism by which you will receive your certificate of attendance, which will support continuing education credits for RAPS, SOCRA, SQA, and ACRP. Once you complete the survey and evaluation, you can then download a certificate of attendance, which may be used in support of these credits. The certificate must be downloaded immediately after completing the survey. Otherwise, the opportunity is lost. Even if you do not want or need a certificate, please take a few minutes to complete the webinar evaluation, as your feedback will help guide the design of our future events. Please note that the evaluation and certificate are only available for 2 weeks, which means the deadline date is March 13th. Now I'll take a moment to call your attention to our upcoming events. On March 13th, SBIA will host the Model Master Files, Advancing Modeling and Simulation in Generic Drug Development and Regulatory Submissions webinar. On April 9th through 10th, SBIA will host the Generic Drugs Forum 2025 conference. We also invite you to visit our CDER Learn webpage, where you can get links to SBIA tutorials along with other educational resources, many of which offer CEUs. That webpage is www.fda.gov/CDERLearn. It is now my pleasure to introduce our presenters. First, we will hear from Dr. Yan Wang, Deputy Director in the Division of Therapeutic Performance 1, or DTP1, within the Office of Research and Standards, ORS, and the Office of Generic Drugs, or OGD, within CDER at FDA. Next, we will hear from Dr. Mark Donnelly, Senior Pharmacologist in the Division of Quantitative Methods and Modeling, DQMM, within the Office of Research and Standards, and the Office of Generic Drugs within CDER at FDA. Next, we will hear from Dr. Qianyan Zhang, Staff Fellow within the Division of Therapeutic Performance 1, within the Office of Research and Standards, and the Office of Generic Drugs within CDER at FDA. Our next presenter will be Dr. Ying Jiang, chemist in the Division of Therapeutic Performance 1, within the Office of Research and Standards and the Office of Generic Drugs within CDER at FDA. Then we will hear from Dr. Pamela Dorsey, Senior Pharmacologist in the Division of Bioequivalence 3, or DB3, and the Office of Bioequivalence, or OB, and the Office of Generic Drugs within CDER at FDA. Lastly, we will hear from Dr. Christopher Morgan, Pharmacologist in the Division of Pharmacology and Toxicology Review, or DPTR, within the Office of Safety and Clinical Evaluation, or OSCE, and the Office of Generic Drugs within CDER at FDA. Please join me in welcoming our first presenter, Dr. Yan Wang.
Hello everyone, this is Ian Wang. I'm the Deputy Division Director for the Division of Therapeutic Performance 1 in the Office of Research and Standards, Office of Generic Drugs. Today, I'm going to present on mastering controlled correspondences, what, when, and how. The learning objective of today's talk is to better understand controlled correspondences related to generic drug development through discussing the role of controlled correspondences, what is the scope and topics that can be included in CCs, and what are common issues and general recommendations for preparing high-quality controlled correspondences. First, I would like to briefly discuss what is a controlled correspondence. CCs are written inquiries to the FDA for information on a specific element of generic drug development or certain post-approval submission requirements. CCs are triaged by the Office of Generic Drugs to a corresponding discipline within OGD or outside OGD. For instance, for quality questions, the CC will be assigned to the Office of Pharmaceutical Quality. What is the role of CC during generic development? CCs are specific inquiries allowing for detailed questions regarding a particular element of generic development, such as formulation, bioequivalence studies, or quality and manufacturing processes. A prospective applicant can obtain early feedback, proactively engage with the FDA to seek input on critical aspects of a development program. Therefore, it improves communication to allow a timely and structured communication channel, fostering a collaborative environment between the FDA and generic industry, leading to clear expectations, which then can be helpful to reduce delays and assessment cycles. By addressing potential critical problems, it can help to reduce the likelihood of delays during development and ANDA assessment, and it can also facilitate preparation of ANDA submission. In today's webinar, you will hear a lot more detailed discussion on specific examples when CCs are helpful during development program. So in this slide, I'm just going to give you a few high-level description on examples when CC may be used. CCs can be used to obtain feedback on the potential acceptability of a proposed analytical methods for testing or characterizing formulations. Can also be used to seek clarification on the design of BE studies, or to discuss potential deviation and or alternative approaches to the recommendations in the product-specific guidances. Can also be used to inquire about the interpretation of a regulatory guidance related to a particular aspect. For example, FDA has published a pH-adjusted guidance for non-Q1, Q2 formulations for ophthalmic otic parenteral formulations, and companies can submit a CC to clarify their understanding and proposed strategy of utilizing the recommendations outlined in the guidance. The CCs are also helpful to discuss the appropriateness of a proposed quality control strategy, stability testing plans, and or manufacturing processes. In this slide, I'm going to discuss a few common issues of CCs that we have seen. The first one is the CC does not contain enough background information and or supportive data to allow proper assessment. Second, the CC is a lack of clarity. The questions are phrased poorly, or in contrary to not having enough information, the CC could contain too much information but irrelevant, or lengthy detailed information without specificity. That could significantly dilute the focus of the inquiry, and it's also hard for us to link the directly relevant information to the proposal to support addressing the questions. The third kind is the CC questions are too general. They are very open-ended. There are certain questions that cannot be directly addressed at the pre-ANDA stage, such as the acceptability of a proposed quality specification. Those are considered as assessment questions, which would require comprehensive assessment of the whole ANDA submission. And another example is the firm sometimes provides a study plan protocol, but they do not raise specific questions. They simply ask if we have any comments on the study plan and protocol. Without any specific questions, we cannot just simply review the protocol and let you know if the protocol is acceptable or not. The last category we have seen is some CCs would have multiple questions, but mix them by crossing different disciplines, such as BE questions mixed with quality questions. So those are some of the common issues of CC that we have seen that presented challenges for us to properly address the CCs. Following the discussion on common issues of CCs, I'm going to provide a few general recommendations for preparing good quality CCs. First, provide sufficient background information and/or supportive data to allow proper assessment. Second, propose concise questions. Raise specific technical and/or regulatory questions. If you are seeking feedback on study protocols, such as alternative BE study design or clinical study design, make sure you raise specific questions. Ask questions seeking for feedback on proposed strategy thinking instead of open-ended questions. Using specification as an example again, you should not ask question on the acceptability of the specification, but it is appropriate to clearly describe your proposed strategy thinking for setting specification, and we can provide feedback on those strategy and thinking to facilitate your development. For Q1, Q2 CCs, provide sufficient information demonstrating your knowledge on the reference listed drug, and clearly describe potential formulation differences if there are intentional formulation changes, such as changes in exception excipients and/or pH adjusters. If Q1, Q2 formulation sameness similarity is required per regulation or is recommended to follow a BE study design, we recommend you resolve the Q1, Q2 issues before proceeding with other related developmental questions. Third, we recommend you separate CCs for different disciplines. For example, you submit separate CCs for BE purpose versus quality purpose. We have been consistently working on improving effectiveness and clarity of our CC responses. Here are a few examples. For Q1, Q2 CCs, we provide concise instruction on the essential information missing in the current inquiry to the applicant to help them prepare the next inquiry to allow proper assessment. When appropriate, such as without disclosure concerns, we provide guidance on potential regulatory pathway to enable filing. We have also reconstructed our responses by clearly indicating whether a proposal is acceptable, not acceptable, or provisional when specific questions are clearly raised. We have also recommended the applicant to submit pre-ANDA development meeting inquiries to further discuss complex questions raised in a CC when we think it's beneficial for the applicant to engage with the FDA to have a multidisciplinary discussion directly. I have talked about the role of CCs during generic development. In this slide, I'm gonna switch gear to briefly talk about how CCs are used within the FDA. The information received in CCs are helpful to evaluate effectiveness, clarity, and relevance of guidance recommendations, including product-specific guidance for recommendations on designing bioequivalence studies or some other general guidances such as pH adjuster guidance, topical Q3 guidances, quality-focused guidances. We also use information obtained through CC to implement continual improvement in regulatory practice, as well as to promote development of new initiatives in both the research efforts and or policy space. So in summary, CCs can be very helpful for both the FDA and generic industry, and we do encourage early engagement through CCs by providing good quality CCs containing specific questions. That concludes my talk. Thank you for your attention. I will be happy to address any questions you may have during the Q&A panel discussion. I will pass the floor to the next presenter, Dr. Mark Donnelly. Thank you.
Good afternoon. My name is Mark Donnelly. Today, I'm going to talk about controlled correspondence on clinical pharmacology topics in generic drug development. Before we begin, this slide is a reminder that the presentation reflects the views of the author and should not be construed to represent FDA's views or policies. The overview of my presentation is summarized here. First, I will provide a brief overview of the controlled correspondence or CC process, including the classification, timelines, and recommended practices with a focus on clinical pharmacology topics. Then I will highlight a few case examples of CCs related to alternative bioequivalence or BE study designs. Finally, I will highlight mechanisms outside the CC process for agency feedback. Before discussing the CC process, I first wanted to briefly highlight some background on our division. In the Office of Generic Drugs, the Division of Quantitative Methods and Modeling evaluate or utilize various modeling and simulation methods and quantitative clinical pharmacology tools to support regulatory decisions and help facilitate generic drug development. The toolkit for modeling and simulation methods encompasses PBPK models for both oral and non-oral drugs, conventional population-based PK and PD models, computational fluid dynamic models, and big data analytics tools such as artificial intelligence and machine learning. To start, the CC process provides a mechanism for timely feedback from FDA on a particular element of generic drug development. As shown here, FDA provides general guidance for industry on controlled correspondence related to generic drug development. As noted previously, this presentation focuses on CCs related to clinical pharmacology topics. Also, it is important to note that some requests may be better addressed through other mechanisms, such as a product development meeting, also referred to as a pre-ANDA meeting, or the Model Integrated Evidence, or MIE, industry meeting. This slide summarizes the types of CC classifications and their associated timelines. A level 1 CC is due within 60 calendar days of the submission date, and level 1 CCs related to clinical pharmacology topics may include guidance clarification or special considerations related to in vivo BE studies. Level 2 CC is due within 120 calendar days of the submission date. And level 2 CCs on clinical pharmacology topics may include evaluations of alternative BE approaches, evaluation of clinical content, or input from another office or center. Here I show examples of level 1 and level 2 CCs. An example of a level 1 CC could be an inquiry to FDA on the acceptability of reporting Cmax and a suitably truncated AUC, such as AUC 0 to 72 hours, as primary endpoints for a BE study with PK endpoints for a drug with a long half-life. In this case, it is considered a level 1 CC because of the request for clarification of FDA guidance and its application to this drug product. An example of a level 2 CC could be an inquiry to FDA on the acceptability of a three-way crossover design to establish BE between the test product versus the US reference listed drug and the European reference product, with additional questions related to the statistical model for analysis. This example would be considered a level 2 CC because of the alternative BE study design and involvement of multiple offices. This slide highlights some potential discussion topics for CCs related to clinical pharmacology. For example, it is common for FDA to receive questions and clarifications related to published BE guidance, such as suitability of AUC truncation and assessment of steady-state attainment in PK studies. Other potential topics may include study design and protocol development, data analysis, alternative BE metrics, and alternative study designs. We don't have enough time to cover these topics in this presentation, but I will highlight a few examples later. This slide highlights some potential discussion topics for CCs related to PBPK modeling and simulation approaches in generic drug development. For oral drug products, CC questions may be submitted for proposed PBPK approaches to support biowaivers of in vivo BE studies or to justify the BE study design, such as a single-sex population. For non-oral drug products, CC questions may be focused on the validation plan of PBPK models for topical, inhalation, and long-acting injectable drug products, or the selection of studies for model development versus model validation. In the next few slides, I will provide a few relevant examples. First, I wanted to highlight the example of adaptive designs for BE studies with PK or clinical endpoints. For some drug products, there are significant challenges with the recruitment of an adequate number of patients for a BE study with PK or clinical endpoints. To address these challenges, applicants have proposed various adaptive designs, including sample size re-estimation at the interim analysis. It is important to note that the recommendations on adaptive design are included in the revised FDA guidance for industry, statistical approaches to establishing bioequivalence. In general, the use of an adaptive design may serve as an acceptable alternative BE study design, but in some requests, there was insufficient information to support the proposed adaptive design, or the proposed methods were not appropriate based on the study design and BE testing recommended in the product-specific guidance, or PSG. In a similar fashion, there are challenges with conducting in vivo BE studies for long-acting injectable, or LAI, drug products. The product-specific guidances for LAI products recommend a single-dose parallel PK study in cancer patients. but the required sample size for the study design recommended in the PSG is challenging for the cancer patient population. As a result, multiple CC inquiries have been received with proposed alternative crossover designs. One of the proposed alternative crossover designs is a single-dose, two-way, or fully replicate crossover design in patients without a washout period. Compared to a parallel design, crossover designs significantly reduce the required sample size, especially with a fully replicate crossover design. This alternative approach may be especially useful for BE studies in certain patient populations with recruitment challenges. However, one concern is that the drug release could extend beyond the dosing interval and impact estimation of AUC. Also, drug carryover may be different between the test product and reference standard. So for applicants proposing this approach, it is important to address concerns of potential unequal carryover effects and its impact on BE assessment. Another alternative study design is a steady-state PK study with repeated crossover design. In this design, 2 consecutive PK measurements are taken for both the test product and the reference standard under steady-state conditions using the sequence TTRR or RRTT. This design can shorten the duration of the study compared to the conventional 4-way fully replicate crossover design using the TRTR or RTRT sequences, because the treatments only switch once. Also, this study design allows for widened BE limits using the reference scaled average BE approach for highly variable drugs. In general, this alternative BE approach may be reasonable, but an applicant should provide proper justification, including sufficient evidence to ensure the absence of drug release beyond 2 dosing intervals. Additional information related to this topic can be found in the poster presentations referenced below. This slide highlights some recommended practices related to CC inquiries as it relates to a specific example. Previously, FDA received an inquiry on acceptability of a PK study for a narrow therapeutic index drug with alternative BE statistical criteria based on outlier data in a PK study. An information request was sent to the applicant to provide additional data and clarifications, but no response was received within the requested timeframe. Here I wanted to emphasize that it is critical to provide all relevant data and information to address the CC requests to avoid information requests from the agency and potential delays. In a follow-up CC, the requester addressed the information request from the agency and proposed an additional question related to exclusion of outlier data in the statistical analysis. In this case, it may have been more efficient to propose multiple questions in a single CC request. As previously noted, some requests may be better addressed through other mechanisms. In October 2023, OGD launched the Model Integrated Evidence, or MIE, pilot program. This is a dedicated regulatory platform to foster early and focused interactions between industry and FDA on MIE approaches for establishing BE in generic drug development. You can find more information about this program in the link below or in the resources slide at the end of the presentation. In summary, the CC process is a mechanism to solicit feedback from FDA on a particular element of generic drug development. Applicants are encouraged to submit CC requests with focus questions related to in vivo BE studies. And this process may support protocol development or evaluation of clinical studies. Also, requesters may propose specific questions related to alternative BE approaches, such as alternative study designs or methods for assessment. Again, we recommend you provide all the relevant data and information to address the CC request to avoid information requests from the agency and potential delays in response. Also, note that certain requests may be better addressed through other mechanisms, such as the product development meeting or MIE industry meeting. And my closing remark is that we recommend you to consider the controlled correspondence process to solicit feedback from FDA on specific questions related to in vivo BE studies to facilitate generic drug development. It provides a list of resources, including relevant FDA guidance related to this topic. And finally, I would like to acknowledge my colleagues in DQMM, ORS, and SBIA who contributed to this presentation and the webinar today. Thank you.
Thank you, Mark, for the great presentation. Today, I'm going to speak to you about the considerations and best practice when prepared for the Q1, Q2 sameness inquiry. Here is the outline of today's presentation. I will talk about the definition of Q1, Q2 sameness and when the Q1, Q2 sameness is involved in generic product development. I will also talk about the best practice for preparation of the Q1, Q2 CC, as well as the different types of FDA response related to Q1, Q2 sameness CC. The first question is, what is Q1, Q2 sameness? Q1 sameness means that the test product uses the same inactive ingredient as the reference listed drug. Q2 sameness means the concentration of the inactive ingredient used in test product are within 5% of those used in ILD product. Next is when Q1, Q2 sameness involved in generic product development. One scenario is when the Q1, Q2 sameness is required by regulation. Per 21 CFR 314.94A9, the drug product intended for parenteral use must contain the same inactive ingredients and in the same concentration as the ILD. Applicant may seek approval of a product that differs from ILD in preservative, buffer, or antioxidant, provided the difference does not affect the safety and efficacy of the proposed drug product. For drug product intended for ophthalmic or otic use, it also should contain the same inactive ingredients and in the same concentration as the reference listed drug. Applicant may seek approval of a product that differs from ILD in preservative, buffer, substance to adjust tonicity, or thickening agent, provided the difference does not affect safety and efficacy. To conclude, the regulation requires parenteral, ophthalmic, or otic use products to be Q1, Q2 the same as the ILD. The prospective and applicants therefore can accordingly submit control correspondence for these products for Q1, Q2 sameness assessment. Another scenario when Q1, Q2 sameness involved in generic product development is when it is recommended by PSG. Here is a snapshot for the PSG for doxycycline cyclate periodontal product. The PSG specifies that to qualify for the in vitro study recommended in the guidance, the test product should be Q1, Q2 the same as the ILD. In this circumstance, although the test formulation is not required to be Q1, Q2 the same as ILD by regulation,
The prospective and applicants can still submit a CC to ask whether the test formulation is suitable for the specific B approach recommended in the PSG. In this slide, I want to talk about the Q1, Q2 sameness assessment. Q1 assessment generally considers the identity of the inactive ingredient. Applicant should provide clear information on the name, grade, salt form of the inactive ingredient. Comparative characterization may be required, which I will provide in detail in the later slides. The Q2 assessment is based on the difference of the quantity between the test and the reference formulation. In this section, I will talk about the recommendation and the considerations when you prepare a Q1, Q2 control correspondence. A Q1, Q2 control correspondence can contain up to 3 test formulations for assessment. Each test formulation should have a well-presented composition table as shown below. For the identity of the inactive ingredient, we understand the ingredients may be referred as different names. In general, the official USP/NF monograph title or the GSRS preferred name should be used in the composition table. The USP/NF title may refer to a family or a product line. Therefore, the grade of the inactive ingredient should be clearly specified. Manufacturing aids that are removed after the manufacturing process and present as trace amount in the finished product are outside the scope of Q1, Q2 assessment. In addition, it should be clearly labeled as if you include the processing aid in the test composition. Another thing is that we understand sometimes it is intended to have certain formulation difference with regard to the exemption excipient. To ensure that the proposed different excipient fall within the list of the permissible inactive ingredient, we recommend you investigate the function of the excipients based on the comprehensive understanding of the formulation. In this slide, I want to talk about the considerations when preparing for the quantity information of the test composition. We want to remind you that the Q1, Q2 sameness assessment is a nominal-based comparison using the composition table with target amounts of inactive ingredients. That is being said, overage is not within the scope of Q2 assessment. Occasionally, due to the potential variability in manufacturing, the actual quantity may be deviated from the targeted amount. If you believe the lactive response for your test formulation is due to this reason, you could consider provide reverse engineering data to support the test formulation in a new control correspondence. As we talked before, the test product should be within 5% difference when comparing to reference product. Therefore, it is important to clearly identify the hydration form for certain inactive ingredients for proper assessment. The units of the marrow for the quantity or concentration of the inactive ingredients should be clearly listed in test composition table. For certain inactive ingredients that may be added on a volume basis during manufacturing, for example, polysorbate 80, please provide the corresponding calculation or equivalent amount, weight-based amount for the ingredients in the test composition. For the test composition table, the total percentage for weight to weight should always sum to 100%. However, for the weight to volume percent in suspension products, the total percentage may exceed 100% depending on the product's density. For example, if a test formulation has a density of 1.05 gram per ml, the total weight to volume should be 105%, not 100%. As mentioned earlier, the Q1-Q2 sameness assessment is a nominal-based comparison. Generally, we do not need characterization data to support Q1-Q2 sameness. In some cases, comparative characterization data on finished product is required to support the Q1 sameness of the excipient. One scenario is that when the starting material do not represent the component in the finished product, for example, the cross-linked enantiomer PDMS. Another scenario is that the property of the excipient may be altered during manufacturing process, and the property of the inactive ingredient is critical to drug release. For example, the algae polymer PLJ or PLA. For algae polymers, comparative physicochemical data on polymer extracted from finished test and RLD products should be provided to support Q1 sameness. The characterization data should include polymer composition, molecular weight, and weight distribution, as well as the polymer architecture. The branch frequency should be provided if it is a branched polymer. For products used a mixture of the algae polymers, to support Q1 sameness, we do not need characterization data of the separated individual PLJ components. Here we are providing some literature for your reference for PLJA characterization. In addition, to support quality assessment of the test product, you should provide characterization data on extracted polymer, including but not limited to polymer endcap analysis, inherent viscosity, and glass transition temperature. This slide, I want to talk about one of the potential pathways for filing ANDA with difference in pH adjuster. We have known that for products for parental, ophthalmic, or optical use, any difference other than the permissible difference will lead to RTR of the ANDA application. We know pH adjuster is not one of these permissible differences. The pH waiver guidance listed over here provides one potential pathway for filing an ANDA with difference in pH adjuster. A test formulation that differs in pH adjuster and without the waiver request for pH adjuster difference is likely to be RTR'd. It is recommended applicants submit a control correspondence to seek FDA's feedback on the proposed justification for pH adjuster difference to support the planned 314.99B waiver request. Whether the agency grants the pH adjuster waiver is an ANDA assessment issue. In this section, I will talk about the FDA responses to the Q1-Q2 sameness assessment. When the test formulation is Q1-Q2 the same as ILD, we generally provide the response that OGD has made a preliminary determination that it would not likely refuse to receive an ANDA application based on your proposed test formulation. If the product is a parental solution, we will also provide a response that OJD would likely grant a waiver of in vivo BE because BE would be self-evident. If the product is not for parental, ophthalmic, or optical use and the sameness, the Q1-Q2 sameness is recommended by PSG, we will provide the response that with respect to the test formulation, OJD would likely recommend the studies described in the PSG to establish bioequivalence. When the test formulation is different to ILD with respect to the exemption excipient, the test formulation is still eligible for filing. Therefore, we will provide the response that OJD has made a preliminary determination that it would not likely refuse to receive an ANDA. However, OJD would not likely grant a waiver of the in vivo BE because BE would not be self-evident. The test formulation is not Q1, Q2 the same as the ILD with respect to one or more inactive ingredients, and we will recommend you use the in vivo BE study to establish BE. When the test formulation is Q1, Q2 different to the ILD, we generally provide the response that OJD has made a preliminary determination that it would likely refuse to receive an ANDA based on your test formulation. Your proposed formulation is not Q1, Q2 the same as the ILD with respect to one or more inactive ingredients. Next, I want to share some useful resources for your generic product formulation development. First of all, the regulation for generic test composition, 21 CFR 314.94A9, 3, and 4. In addition, here is a list of guidance for industry may be beneficial for your generic formulation development. We have guidance for submitting control correspondence to FDA requesting information related to generic drug development. We also have guidance describe best practice for writing the composition statement. In addition, as mentioned in the talk, we have the guidance for pH-adjusted vivo for parenteral, ophthalmic, and otic use drug products. We understand the ILD labeling is generally used as a starting point for generic product development. The 21 CFR 201.100 Section B provides the requirement for the information to be included in the labeling. You could also check the most recent labeling using the website provided below. Last but not least, OJD has a lot of GDUFA research for reverse engineering for certain complex products. You could refer to the publication for your formulation product development. To conclude, Q1, Q2 sameness refers to the same inactive ingredients, the identity, and the amounts each within 5% to the ILD. The provide a test product composition table should have clear identification of inactive ingredients and accurately list the amount for each inactive ingredient for proper Q1, Q2 assessment. To support the Q1 sameness of the algae polymer, comparative characterization data on the polymer extracted from finished product should be provided. If you are going to submit a 314.99B vivo request in your ANDA application for the pH-adjusted difference, it is recommended you submit a CC to seek FDA's feedback on the proposed justification for pH-adjusted vivo. Whether the potential vivo request can be granted is an ANDA assessment issue. Thank you for listening. I will be back at the QA session for any of your questions. Then I will pass the floor to my colleagues to talk about consideration for products that are not required to be Q1, Q2 the same.
Hello everyone, this is In Zhang. Thank you very much for the earlier introduction. Today I'm going to talk about the general considerations and best practices for getting feedback related to inactive ingredients or formulations, and I will be focusing on the generic drug products that are not required to be Q1, Q2. In this talk, I'm gonna cover when it may be beneficial for you to submit an inquiry related to formulation during your generic drug product development. I will also cover what information to be included in a control correspondence to avoid pitfalls that may cause a delay of an assessment. For the purpose of today's talk, I'm going to use topical products as an example. The 21 CFR says that for topical generic drug products, they may include differences in inactive ingredients compared to the ROD, as long as the differences are identified and characterized and appropriate justifications are provided. So in other words, a topical generic or test product is not required by regulation to be Q1 and Q2 the same as the ROD. Typical B approaches for topical products include a comparative clinical endpoint B study, vasoconstrictor B study, waiver of in vivo B study, and characterization-based B approach. Depending on the scientific thinking behind the approaches, some approach may include a formulation standard within it, like the characterization-based B approach. We'll talk about the formulation assessment under the characterization-based B approach in a little bit. However, before we go there, I want to emphasize that regardless of the B approach you decide to use, there is a universal expectation that you need to ensure the proposed level of use for inactive ingredients are acceptable, which would lead us to the discussion about maximum daily exposure assessment. Information available in the inactive ingredient database for approved drug products can be useful to guide your formulation development. However, it does not have all the information necessary for an analysis or comparison of the context of use. The route of administration is listed in the IID. However, the duration of use or patient population are not included in the IID for those records. So if you do have a question related to MD assessment for your proposed inactive ingredients, we strongly encourage you to submit an inquiry early in the product development, ideally before you conduct any costly and time-consuming studies with a potentially problematic formulation. And this is a list of guidances for industry that may help you prepare the content and format of such an inquiry. Next, let's talk about the formulation assessment under the characterization-based B approach. This is a frequently asked topic within the CC program. Characterization-based B approach exists in PSGs for many topical products. It was developed based on the principle that a test product would be designed to be closely matched to the RS, just like the RS would be matching itself from batch to batch. So this B approach does kickstart with a formulation standard, which is a no significant difference or NSD standard. And this approach also includes other comparative studies to ensure that the proposed test product also has the same microstructure or Q3 or performance as the reference product. So how does the NSD standard compare to the Q1, Q2 Sameness standard? The NSD standard is built upon the principle for Q1, Q2 assessment. So a Q1, Q2 formulation is a sweet spot under the NSD assessment. Certain differences that may not be acceptable under the Q1, Q2 Sameness criteria may found to be acceptable under the NSD assessment criteria based on available scientific evidence. Below shows how the NSD standard and Q1, Q2 example are described in the PSG page. Also, you can tell from this PSG language that under NSD assessment, it's possible to compare the proposed test formulation against the reference standard, even if it may not be the same product as the ROD. With all that, I hope you get the sense that the NSD standard really does not mean that any formulation would be acceptable. Here comes an example of an NSD product. I'm going to talk about just 2 inactive ingredients in the interest of time. This is an inactive ingredient with different hydration forms. So it is acceptable to use a different hydration form as long as the amount of the anhydrous form match between the test and reference product. For pH adjusters, more than 5% differences are generally acceptable if the pH of the final product are matched between the test product and the RS. To stretch that a little bit, it's even acceptable to list QS to target a certain pH or pH range for the test formulation. So how do you get feedback for an SD assessment? All begins with asking the correct question. What we are able to address is whether one or more proposed formulations may be eligible for the specific BE approach that you're interested in. So do ask that. If you ask whether the proposed formulation is Q1, Q2 the same, your CC may get declined for substantive review. In the CC submission, try your best to include all necessary information related to the formulation. Missing information that impacts the assessment will call for an information request. That essentially means an extension of go date. Going into more details about the information to include in the NSD assessment submission, from Q1 perspective, we recommend you to include the correct compendial grade and the name. If there's subtype under monograph, you may want to specify the subtype as well. As an example, for carbomer homopolymer, you may want to specify whether it's type A, type B, or type C. You want to specify the salt form or hydration state for the relevant ingredients. If you list a proprietary name, make sure you list a full name and not a shortened one, because the shortened one may relate to several excipients and they may have different compositions. To give you an example on that, if you list quota for CES, we may have to send an IR asking what is the full name and what is the composition for the excipient you are proposing. Sometimes a certificate of analysis may be helpful in the submission. From a Q2 perspective, we recommend a minimum of 2 decimal places for each inactive ingredient in the relevant units. If your test product happens to include alcohol, sometimes it's helpful to include the absolute amount of alcohol within your proposed test product as well. That can assist the assessment. For inactive ingredient to be added on a QS basis, make sure to specify the target value, for example, the pH or the pH range. If you propose an overreach for an inactive ingredient, make sure you clarify what is the nominal amount without the consideration of that overreach, because only the nominal amount will be considered during the Q2 assessment. If you need to explain any Q1 or Q2 aspects, you can include reverse engineering data in your submission as well. A couple more considerations to mention here. For mixture and active ingredients, we recommend you to include what are the components, what are the proportions of the components, and how it is manufactured in the CC submission. The manufacturing method has something to do with the Q1 acceptability. For example, for a co-processed mixture, you may not replace it with separately added excipients. However, for physical blend, you can do so. Here I'm showing an example of the combo of microcrystalline cellulose and carboxymethyl cellulose sodium. The separately added combo of these two excipients do not equal to the co-processed one from Q1, Q2 perspective. This excipient combo may be more relevant for the nasal and inhalation products. I want to note that. For compounding kit product, if you are proposing one, we encourage you to provide formulation composition tables for both before and after mixing, each bit on a 100% basis. The only providing the amount in the final compounded product may not be sufficient sometimes. So that pretty much covers everything about the formulation assessment and the characterization based B approach I want to talk about. And here I want to briefly touch upon the possible feedback you may get for the topical and transdermal delivery systems or TDS products. In the PSGs for this category of products, we usually recommend 3 in vivo B studies: PK study, adhesion study, and irritation and sensitization study. If you have not noticed this, I want to bring to your attention that as part of the general guidance for the skin irritation and sensitization study for ENDAs, it is noted that sometimes the sensitization evaluation may not be necessary if adequate justification is provided. So if you want to explore this, you can submit an inquiry with information about your product formulation, your product design, and any relevant justification you may want to include to support your position. In summary, engaging with the agency through the CC program to gain feedback on formulation aspects can be beneficial for your product development program. And we strongly encourage you to try your best to provide all essential information needed for the specific product and the B approach. This will help enhance the efficiency in processing and assessment of the CC. With that, I would like to thank all of these FDA colleagues for their contribution or support to the preparation of my talk. Thank you very much for your attention as well. I will be back for Q&A towards the end. Next, I would like to turn it over to the next speaker, Dr. Pamela Dorsey.
Good afternoon. My name is Dr. Pamela Dorsey, and I am from the Office of Bioequivalence. My presentation is entitled Bioequivalence Considerations for Control Correspondences, Assessment and Best Practices. Within this presentation, I will describe varying types of control correspondences assessed by the Office of Bioequivalence, or OB. Secondly, I will highlight how to differentiate between a Level 1 and Level 2 control correspondence, or CC. And finally, I will provide examples for 3 categories of CCs that we assess regularly. These include evaluation of maximum daily intakes for inactive ingredients, the use of a reference standard that is not listed in the Orange Book, and bioequivalence documentation needed for prior approval supplements. Let's take a deeper look at types of controlled correspondences assessed by the Office of Bioequivalence. OB is responsible for assessing the following categories of CCs. These categories include the addition of a new strength to an ANDA, evaluation of proposed maximum daily exposures for inactive ingredients, alcohol dose dumping questions, the use of a reference standard or RS other than what is currently listed in the Orange Book for that drug product, questions related to skin blanching and vasoconstrictor studies, cross-referencing bioequivalence or BE studies, retention sample recommendations, comparative dissolution testing, pre or post-approval changes to an ANDA, and questions regarding study conditions and or design. Please note that this is not an exhaustive list. Within the category related to the use of reference standards, which are not listed in the Orange Book, these CCs can contain questions regarding the use of authorized generics, the use of the RLD, which is not listed as the current RS at the time, or use of a different strength, which is not currently recommended by the product-specific guidance, or PSG for a drug product. Within pre- and post-approval changes, OB can assess questions related to OB documentation needed for API source changes, formulation changes, and manufacturing site changes. As a miscellaneous category, OB will also assess questions regarding the number of lots needed for in vitro studies, protocol review for covered product authorizations, post-CRL questions, and PSG teleconferences. Now let's switch gears to determine what distinguishes a level 1 CC from a level 2 CC. A level 1 CC is referred to as a standard CC, whereas a level 2 CC is referred to as a complex CC per the GDUFA 2 commitment letter. CCs that receive a level 2 designation automatically include questions regarding evaluation of clinical content, covered product authorizations, and alternate BE approaches. The controlled correspondence related to generic drug development guidance states that level 1 CCs are to be reviewed in 60 calendar days, and level 2 CCs are to be reviewed in 120 calendar days. Please note that level 1 CCs can be switched to level 2 CCs if input is needed from other offices within the agency to answer the question. For the next section, let's explore examples of categories for CCs commonly assessed by OB and best practices for these categories. The first commonly received CC category that OB will assess are questions regarding the acceptability of proposed maximum daily exposures, or MDEs, for inactive ingredients within an ANDA formulation. Please note that MDEs for inactive ingredients are evaluated in all populations for which the intended drug product is indicated for, unless an applicant specifies a specific population in their cover letter. Proposed MDEs are justified based on MDEs for the same inactive ingredient within previously FDA-approved drug products, either NDAs or ANDAs, for the same route of administration and similar context of use. I would like to highlight that it is not recommended that clinical or toxicological data be submitted with your inactive ingredient CC, as that is outside the scope of review. When submitting CCs regarding the evaluation of proposed MDEs for inactive ingredients, please remember the 3 and 3 recommendation per the controlled correspondence related to generic drug development guidance. To align with the guidance, it is recommended that the number of evaluations not exceed 3. Therefore, submit 3 inactive ingredients with 1 proposed amount each, or 1 inactive ingredient with 3 proposed amounts each. In this context, the amount means the amount of the inactive ingredient per unit, with the corresponding MDE calculated by using the maximum daily dose of the drug product. One evaluation is also applicable to drug products that can be given via different routes of administration. Evaluation of the inactive ingredient, NDE, for each route of administration corresponds to one evaluation each. The same recommendation applies to a drug product that has varying dosages depending on weight, age, or even body surface area. Evaluation of an inactive ingredient for each range constitutes one evaluation. Additionally, the acceptability of components within an inactive ingredient that contains multiple components, such as a flavor, does not guarantee the acceptability for the inactive ingredient as a whole. Let's discuss our first example. In example number 1, a CC was received for the evaluation of a proposed MDE for disodium hydrogen phosphate dihydrate and phytonadione injectable emulsion. This drug product can be administered via IV, IM, and sub-Q in the adult and pediatric population per the RLD label. To comply with the 3-in-3 recommendation, the proposed MDE for disodium hydrogen phosphate dihydrate was evaluated in the adult population for the IM, sub-Q, and IV routes of administration in adults. The applicant was asked to submit separate CCs for the evaluation of disodium hydrogen phosphate dihydrate in the pediatric population for each route of administration, as the evaluation of the MDE for disodium hydrogen phosphate dihydrate in the adult and pediatric population for all routes of administration would exceed 3 evaluations. For example number 2, an applicant has submitted a CC for the evaluation of one proposed amount for sucrose, xanthine gum, and polysorbate 80, respectively, in ibuprofen oral suspension. Ibuprofen oral suspension is indicated for children 2 to 11 years of age, and the dosage varies within the pediatric population based on the weight and age of the child, as seen in the dosing table. 3 inactive ingredients, with one proposed amount each and 5 different dosage ranges, would exceed the 3-in-3 recommendation. Therefore, the proposed amounts for sucrose, xanthine gum, and polysorbate 80 were evaluated for the 2 to 3-year-old range in the pediatric population, as this equates to 3 evaluations per the control correspondence related to generic drug development guidance. The applicant was informed to submit another CC for the evaluation of the proposed inactive ingredient amounts for other dosage ranges. The second category for CCs commonly evaluated by OB would be the use of an RS that is not listed in the Orange Book. The reference standard is a drug product selected by the agency to demonstrate bioequivalence for an intended ANDA. In most cases, the reference listed drug, or RLD, is usually the RS. However, if the RLD is not available, a previously approved ANDA can be designated as the RS. The designation of an RS is handled by the Division of Orange Book Publication and Regulatory Assessment. However, there are cases where the current RS is not available on the market. In these circumstances, the Office of Bioequivalence may suggest another approved ANDA to demonstrate bioequivalence or provide an alternate BE approach. An alternate BE approach may be appropriate for drug products in which the RLD is discontinued and there is no other approved ANDAs available. Let's talk about our third example. In example number 3, an applicant sought to develop primidone oral suspension. However, the RLD was discontinued and there are no approved ANDA primidone oral suspensions. The applicant submitted a CC asking for an alternate BE approach by comparing their proposed primidone oral suspension to primidone immediate release tablets in order to demonstrate BE via in vivo studies. The agency determined that a PK bridge could be drawn between the tablets and the oral suspension, utilizing information in the new drug submission. Subsequently, the applicant was informed that the alternate approach was reasonable to demonstrate BE. The third category of CCs commonly assessed by OB would be changes to an approved ANDA via prior approval supplements. These changes can encompass a change in manufacturing site, formulation, API source, or the addition of a new strength. For solid oral dosage forms, the SUPAC-IR and SUPAC-MR provide recommendations for bioequivalence documentation needed to support changes to manufacturing sites or the formulation. The SUPAC-SS provides recommendations for non-sterile semi-solid preparations. The level of change will determine whether an in vivo BE study or other studies are recommended. Recommendations for other products that do not fall under the guidances mentioned are product-specific and may require other studies to support proposed changes. Please be aware for changes that necessitate an in vivo BE study, the current reference standard is recommended, rather than comparing pre versus post-change drug product. Further, the conditions under which an in vivo study should be conducted can also be confirmed via a CC. Let's look at example number 4. In example number 4, an applicant company A sought the addition of a 1 gram strength to an approved ANDA which already has an approved 500 milligram and 750 milligram strengths. The RLD only has a 500 milligram strength and a 750 milligram strength, which are both discontinued. Company B has an approved 750 milligram strength ANDA, which is the current RS. However, Company C has an ANDA with a 1 gram strength, which was approved via suitability petition. The current PSG recommends a fasting study utilizing the 750 milligram strength with a biowaiver for the 500 milligram strength. The current applicant, Company A, requested a biowaiver for the 1 gram strength in their CC. However, the biowaiver was deemed unacceptable, and an in vivo study was recommended comparing Company A's intended 1 gram strength to Company C's approved 1 gram strength. So let's review 2 challenge questions. Challenge question 1, which scenario counts as one evaluation for an inactive ingredient control correspondence? A, an MDE from a dosage range based on weight, B, an MDE from a dosage range based on age, C, an MDE for a single component of an inactive ingredient, or D, all of the above? The answer is D, all of the above. Challenge question 2, true or false? The RLD is discontinued and the current RS is not available on the market. I should petition the Office of Bioequivalence to designate a reference standard. False. The Office of Bioequivalence does not designate or assign reference standards in the Orange Book. However, the Office of Bioequivalence can suggest a reference standard to determine BE or provide an alternate BE approach. To request reference standard designation, please submit a CC to the Division of Orange Book Publication and Regulatory Assessment. In conclusion, I would like to highlight the following for your consideration. Please remember the 3 and 3 recommendation when submitting inactive ingredient CCs. Additionally, alternate BE approaches may be possible for drug products given the RS, whether it's the RLD or an approved ANDA, is not available on the market, and there are no other approved ANDAs for BE determination. And finally, prior approval supplements for changes that necessitate an in vivo BE study should utilize the current RLD or RS. A comparison of the pre-product versus the post-product for in vivo BE studies is not recommended. For your reference, I have provided a few resources that are helpful regarding bioequivalence questions within CCs. And with that, I would like to thank the following individuals for their contributions to this presentation. Thank you for your attention, and I will now turn the webinar over to Dr. Christopher Morgan.
Good afternoon, everyone, and thank you to Pamela and the previous speakers. My name is Chris Morgan. I'm from the Office of Safety and Clinical Evaluation, or OSCE. Today I'm going to be discussing the role of controlled correspondences in supporting the safety assessment of generic drugs. As a brief overview, I'll begin with an introduction to OSCE, including who we are and what we do. Then I'll discuss the role OSCE plays in addressing controlled correspondences, or CCs, to support generic drug development, including some of the common CC topics that we address. OSCE is comprised of a multidisciplinary staff organized into 3 divisions, each of which play different roles in supporting the generic drug program. These include the Division of Pharmacology and Toxicology Review, of which I'm a part, the Division of Clinical Review, and the Division of Clinical Safety and Surveillance. Our mission is to ensure that the American public has access to safe generic drug products that are therapeutically equivalent to reference-listed drugs. To fulfill this mission, OSCE evaluates clinical and non-clinical information in support of safety and therapeutic equivalence. We ensure compliance with any safety requirements, such as risk evaluation and mitigation strategies, and we're involved in conducting ongoing safety surveillance after approval. OSCE is involved throughout the life cycle of generic drug products, including in the pre-submission development phase, as well as in the pre- and post-marketing phases. As part of this work, we respond to controlled correspondences that are submitted throughout this same life cycle. Just as a few brief introductory points, it's important to note that controlled correspondences that require the evaluation of clinical contents are categorized as level 2, which means that they have 120-day response timelines. This includes both controlled correspondences that are directed specifically to OSCE, as well as those that are directed to other offices but then require our input. Importantly, a complete safety assessment is outside the scope of a controlled correspondence. Instead, our response will provide guidance, providing an opportunity to gain alignment with the agency's current thinking on a topic, or to receive feedback on how to resolve a deficiency or improve the quality of submission, with the desired end result of reducing the number of assessment cycles required for an application. Some of the common controlled correspondence topics that we address include those that require input regarding the context of use, which includes dose, route of administration, duration of use, and patient population. These are often in support of another discipline's CC responses I discussed earlier. We address those that ask for feedback on approaches to justify the safety of excipients, to qualify the safety of impurities, including process and degradation impurities, extractables and leachables, and nitrosamines. Additionally, we handle controlled correspondences that are part of covered product authorizations. These are requested as part of a process through which generic drug developers can obtain samples of an RLD product for things like bioequivalence studies, when that product is subject to risk evaluation and mitigation strategies that restrict its distribution. I'm not going to be discussing these CPAs further in this presentation, but there will be a link to the applicable guidance at the end of the slides. The safety and quality of a drug product is assessed in the context of its clinical use. Therefore, an accurate determination of the dose, route of administration, and treatment duration, as well as the indicated patient population, is essential in guiding the development of a generic drug product. Some of the controlled correspondence topics that we address related to clinical context of use include determining what the appropriate maximum daily dose, or MDD, is, as well as this is important in determining the resulting maximum daily exposure to any excipients or impurities. We provide feedback on how the route of administration might impact the acceptability of a proposed safety endpoints. For example, the importance of performing a local toxicity study for a drug that's administered topically. We provide feedback on the appropriate duration of use for a drug product over a patient's lifetime, as well as establishing whether or not a drug product is administered during sensitive developmental windows or administered to patient populations that have specific safety concerns that should be considered assessing the safety of a drug product. little bit more information on maximum daily dose and maximum daily exposure. MDD is the highest dose of a drug substance reasonably administered in a day. It's determined based on the dosage and administration section of the reference listed drug labeling. The safety of product formulations is assessed based on the exposure to excipients and impurities at MDD. This is the maximum daily exposure to these excipients or impurities. And applicants can submit a controlled correspondence for drug products with complex dosage and administration instructions, those that are unclear, particularly in the event where a drug product has an atypical route of administration. For example, in some cases, dosing might be based on the body weight or body surface area. As a side note, we generally recommend using standard values of 60 kilograms or 1.62 meters squared for adults, unless for some reason the patient population that's indicated for a drug product falls outside of the normal range. In some cases, a drug might be recommended to dose to effect, such as with anesthetics. And again, there are many cases where a drug product has an atypical route of administration, for example, with a topical drug product. An example of something we might provide feedback would be on an applicant's proposed MDD for a drug product with labeling that simply recommends it be applied as a thin film to affected areas. Excipients are inactive ingredients intentionally added to a drug product formulation that are themselves not intended to exert a therapeutic effect, but may improve product delivery. And safety justification is needed when the proposed exposure to an excipient exceeds the level of that excipient in other products with a similar context of use. Common excipient-related CCs that we address include whether or not an entry in the inactive ingredient database, or IID, is appropriate to justify the proposed MDE to an excipient in a new drug product. In other words, whether or not these two drug products share the similar context of use. We can provide feedback on whether the safety data for one grade of an excipient can be used to justify the safety of another in what we would call a bridging argument. And we can provide feedback on whether or not a proposed approach to justify the safety of an excipient is reasonable. Some of the information used to justify the safety of excipients includes the exposure to these excipients endogenously, perhaps in our diet, as well as in published safety data. Impurities may arise in the manufacture or storage of a drug substance or drug product. Drug substance or product impurities need to be qualified for safety when the proposed exposure to these exceeds applicable thresholds. These thresholds are allowable limits generally established by FDA or ICH guidances, and the specific limits depend on the context of use of the drug product, with exposure beyond these limits often requiring additional safety assessment. Common impurity-related controlled correspondence topics we handle include essentially what are the applicable safety thresholds for a drug product. For example, the M7 threshold of toxicological concern, which is used for potentially mutagenic impurities and is based on duration of use, as well as Q3A or Q3B qualification thresholds for non-mutagenic impurities, which are primarily based on MDD. We address whether or not a drug product falls outside the scope of a guidance. For example, drug products used to treat advanced cancer indications, or drug products that are peptides or oligonucleotides or fermentation products. And finally, we provide feedback on proposed approaches to qualify an impurity that exceeds the applicable thresholds. These can include studies such as comparative impurity analysis, metabolite justifications, mutagenicity assessments, and general toxicity studies. Extractable and leachables. These are compounds that can migrate from manufacturing equipment or container closure systems under exaggerated conditions or over the shelf life of a drug product. Common CCs that we handle related to extractable and leachables include determining what the appropriate safety concern threshold, or SCT, is for a drug product, which is used to inform the analytical evaluation threshold, or AET. The SCT is the threshold above which the safety of a particular compound would need to be justified. It's established based on the route of administration and treatment duration. And then the AET is the threshold above which these compounds need to be identified. And it's determined based on SCT and MDD, or maximum daily dose. We also provide feedback on proposed approaches to justify the safety of the worst case exposure to extractables and leachables, which can include mutagenicity assessments and or general toxicity studies. And finally, as is probably not unexpected, a large proportion of the controlled correspondences we've been handling in the last few years are related to nitrosamines. These are small molecule or nitrosamine drug substance related impurities, and the FDA has published several guidances on the control of these impurities in drugs and approaches to determine the acceptable intake, or AI, limits. Common CC topics that we address for nitrosamines include whether or not a carcinogenic potency categorization approach, or CPCA-based AI limit, is acceptable. In other words, whether or not an applicant has come up with the appropriate potency category. We can provide feedback on whether the proposed approach to justify a higher than the FDA recommended AI limit is reasonable. These might be justified by surrogate-based AI limits, data from enhanced Ames assays, in vitro metabolism, or in vivo mutagenicity studies, including providing feedback on protocols for some of these complex studies. And we should note that we are aware that the FDA recommended AI limits can differ from limits published by other regulatory agencies. But in these cases, we do strongly recommend that applicants apply FDA recommended limits. To quickly summarize what we've gone over and some of the key points. First, OSCE works to ensure generic drugs are safe and therapeutically equivalent. We offer multidisciplinary expertise on clinical, pharm tox, and post-marketing safety surveillance related topics throughout the life cycle of generic drug development. Controlled correspondences that need our input are generally classified as level 2 with a 120-day response timeline. Controlled correspondences provide an opportunity to receive feedback on the context of use, proposed approaches to justify the safety of excipients and impurities, and covered product authorizations. CCs also provide opportunities to align with the agency's current thinking, to improve submission quality, and to obtain feedback on approaches to resolve deficiencies. And finally, and importantly, CC responses are not themselves a safety assessment. And the acceptability of a formulation or justification, a safety justification, is going to be determined during the technical assessment of the ANDA submission. Here's a rather extensive list of resources that we think are useful, covering many of the topics that I've discussed. At that point, I'd like to thank you for your attention, and I look forward to addressing any questions you may have during the Q&A session immediately following the discussion panel, which is next.
Thank you again to our presenters for their excellent presentations. We will now begin our discussion panel. Please join me in welcoming today's moderator, Dr. Priyanka Ghosh, lead pharmacologist within the Division of Therapeutic Performance 1, or DTP1, within the Office of Research and Standards, and the Office of Generic Drugs within CDER at FDA.
Hello, Corey. Thank you so much. And thank you to everyone for joining our panel discussion today. Please join me in welcoming our panelists for this webinar. And to the panelists, I request if you can please turn on your cameras as we go through the introduction. So joining us today, we have Dr. Robert Leinberger, the Director of the Office of Research and Standards. We have Dr. Partha Roy, the Director of the Office of Bioequivalence. And we have Dr. William Chong, the Director of the Office of Safety and Clinical Evaluation. So thank you so much to all of you for joining us today. So we heard a lot during the webinar about some of the specific types of CCs that are handled by the different offices and how getting clarity on these CCs can help towards the ANDA review processes. We were wondering, what are some of the areas that you see from your perspective where getting clarity through the CC process can help with the efficiency of the ANDA submission as well as the review process at this time? And we can start with Euro.
All right, thanks, Priyanka. I think the places where I see the most value of this are things that you want to clarify before you do incredibly expensive bioequivalence programs. Like, for example, our control correspondence lets you get feedback on your drug device combination product. You don't want to go ahead and conduct a study using that product and then you find out later that you have feedback on the device itself and you change it and then you invalidate the study that you did, or you cause questions on how do you bridge between the device you used in the study and the device you want to market. So I think one really valuable thing is for more complex development programs, really getting the simpler things correct first before you do the more intensive parts of the of the study. Instead, before you conduct the BE study, make sure that your formulation is acceptable. Don't go over the maximum daily dose in your formulation, and then you come back and you get comments, can you please reformulate for safety reasons? And then you say, well, what do I do with my BE study that I did on the old formulation? So I think there's a lot of ways that for multifaceted development programs, using this to nail things down before you go into the more expensive studies that you do can create great value for product developers.
Thank you, Rob. Partha, would you like to go next? We cannot hear you yet.
Can you hear?
Yes.
Oh, you can hear me. Okay, good. Yeah. So the other important part.
So Partha,
okay.
It's going in and out a little bit, so I was not sure if there is something we can do to. Maybe I'll go to Bill and come back to you.
Thanks, Priyanka. Hopefully my audio is better. Good. Okay. I think I would agree with Rob that I think there's definitely some things that getting advice and clarity early is important, especially in designing the direction of the development program. I think we heard some really great examples today around the importance of getting a formulation kind of in the right direction. The feedback that can be provided in a control is really critical in identifying the approach to bioequivalence, making sure that you're on the right track for even getting through filing when you submit your ANDA. I think there's lots of other sorts of feedback that are important, and as Rob pointed out, I can imagine that if, as a generic drug developer, before I invest money in doing either a large BE study or developing in vitro approaches to demonstrating bioequivalence, having more certainty in the direction and that I'm starting with a product that seems to be eligible for whatever pathway I'm taking is important, because the last thing that I would want would be to get a letter saying I have to go back, change my formulation, and potentially have to do everything over again. I think there's a couple other areas. We didn't get a chance to talk about it today, and it wasn't really part of the focus, but I know there was a controlled correspondence called a covered product authorization, and that's incredibly important for people who are developing products that are subject to a REMS. That's a pathway by which and the applicants and the developers can get access to our reference products so they can do their testing. I have to imagine it's hard to do a comparative study if you don't have something to compare against. So I think that's another one that wasn't really covered today, but it is an important part of the controlled correspondence pathway. Maybe I'll pause here and see if Partha has his audio working today.
Yeah, can you hear me now? Better? Okay. For some reason, Priyanka, I can't hear you. Okay, that's okay. What I wanted to say is that also the timing is important, right? Because a lot of times I hear that, oh, I want a response, very quickly, right? And I think today you heard about something level 1, level 2, but for all of the bioequivalence questions, we try to keep it within the level 1 and try to have a one discipline response. And I don't want you to kind of caught up in this within the Office of Generic Drugs. We collaborate a lot within each offices. So our intent is to sort of give you the response, effectively and quickly. And I think that's important. But again, what we expect you to do is to give us questions which is more clear, more concise and precise. Because what happens sometimes you ask questions and we may have like 5 different ways of answering the question because we kind of don't understand the context of it, right? So I think that's important to kind of lay out the context, and that becomes extremely valuable. Now, sometimes it happens that, we might have to say that, it cannot be answered to controls. You may move on to the pre-ANDA meeting package because there you actually can lay out your development, because then, so that part becomes very useful. So I think, how to ask the question, exactly what to target, and give us a perspective, I think is important. And that way it really helps us to answer the questions, quickly. So I'm sort of thinking about more the timing aspect of it, because I know that many of you are sort of, running against time. So I think, and we recognize that, and that's where I think it creates a lot of value.
I think it's a perfect segue then, Partha. So one of the things, maybe we identified a couple of questions from the questions we received, and we wanted to see like what in your recommendation would be some ways to improve these questions perhaps, or at least provide clarification so that our assessment teams can provide efficient and useful responses. So the first one indicates that our company wants to use a different inactive ingredient compared to what is used by the reference product. We want to include a nanoparticle inactive ingredient in the formulation, and then it asks what pathway to approval must we follow. I interpret this as a pretty open-ended question, but we would really like your input on like how to make this question actually efficient and useful. And any of you can go first.
I mean, so one aspect of the question of a question that you can use the control correspondence for is, and I think is early in development, is what I'm proposing appropriate for, if it's not, I'm not sure if this is appropriate for an ANDA pathway. I mean, that touches things like the Q1, Q2 is the most obvious one that we sort of routinely do, thousands of them every year, right? Is my, for products that are required by our regulations to be Q1, Q2, you're asking, is my proposed formulation acceptable to come in? That's probably the most common thing that we do. But there are other variations of that in other cases. If you're saying like, look, I have some other something that's not so obvious as to, is this acceptable in this ANDA, and sometimes you can get advice. It might require, something might be larger, it might require a suitability position, it might not be acceptable. But those are questions where you might come in. So if you say, well, maybe I don't know what FDA thinks about nanomaterials, right? I think maybe FDA thinks that they're magically different active ingredients, and I want to check and see, you can ask us that. You can also read our guidances and probably get the answer to that as well. So that, so like early in the development process, you might ask the question, is what I'm proposing really going to be acceptable in the generic drug program if it's something that's there's some difference that I've noted between my product and the reference product. That's something you can do very early in development. As you get later in development, then I think control correspondences are more about the specific studies, right? What type of bioequivalence study I do, what type of bioequivalence study is appropriate for a particular formulation difference, right? And you can also use control correspondences this gets to the ones talking about level 2, to talk about differences. Oh, I want to do something different than your guidance because I have a new idea, or I have a different approach, or I have a different situation. So all of those things can come in if you're doing something, if you have something that you think is possible to be a generic, but maybe not as clearly exactly identical to the brand product. Those are areas where the control correspondences can help you get clarity on whether that's an appropriate idea or not. As well as more complicated situations. If it's for complex generics, you have more access to the pre-ANDA meetings as well to address those more complicated situations under certain situations.
Bill, you wanted to add to that response?
Well, I don't know what I can add to Rob there. He covered a lot. I do think that, for that particular sort of phrasing, I can see how our assessors, when they're looking at that sort of question, might be scratching their head a little bit. I think, simplifying the question, being more straightforward, like depending on where you are in development, if your question is, I have this formulation difference, I want to see if I'm eligible for a certain approach to bioequivalence. That is a little bit clearer, I think, to me at least, that, you could, if you a little bit more succinct, phrase it in a way that's more direct, that might be easier for our folks to get you a response, potentially even sooner than the goal date. But I think that, if you know what your concern is around the difference, then just pointing to it and being clear, saying, we are using this particular inactive ingredient. We know it's not in the RLD. Would we still be eligible for, I don't know, option 1, 2, or 3 in the product specific guidance or something like that? I think that might be a little bit more of a direct question. I think we heard that as some early tips and tricks at the outset of the webinar, sort of on how, being clearer so that we don't have questions about what the question is, can be helpful.
Yes, if I can add just one line, I think we want to understand the context, like why are you using that particular inactive ingredient, or tell us what is bothering you while you're asking the question, because that's an important, right? I mean, what product? Obviously, you'll tell us what product you're using. You probably think that's an inactive ingredient. Maybe you have information to see maybe that inactive ingredients has some effect on the activity of the API. I mean, so those are some of the questions if you have in your mind. Providing that clarity or the context, that really helps us actually, because not only we want to answer that question appropriately, but also if we understand exactly what is sort of why you're asking that question and what is really the intent, then we can actually talk to some of the experts and get more clarification before we answer that question. So I think that's all. So it's better to kind of providing that context and clarity, I think, becomes important. So kind of help you answer the question.
For the excellent recommendations for how to make that better. I'm going to quickly ask a small question that I saw multiple times in the Q&A box, and then go on to the next question on our discussion topic. There were multiple questions about what is the fee for a controlled correspondence and how to submit it. I think that for the latter part, there are guidances, but the first part, if one of you could answer that question.
Yeah, so there's no fee for controlled correspondence. You have to be an appropriate engagement. To be engaged in generic drug development, and have a U.S. aid. Only U.S. entities. So if you're outside the U.S., you must have a U.S. agent. But the controlled correspondence, there's no fee for them. But there is no such thing as a free lunch. The resources that go to support the controlled correspondence program come from the GDUFA fees that applicants pay. Right, so that's So that's, there are people, lots of people who work on this, and that work is supported by the fee paying parts of the program. And that's why the control correspondence questions are focused and intended toward potential applicants, because the applicants are the ones that support the resources that go into the program. But there's no specific fee per question. But the control correspondence are really valuable, so they cost a lot of effort to do. So don't think they're free.
Moving on to our next discussion question. Control correspondences, complex control correspondences versus pre-NDAs. Both of them, give or take, have a 120-day goal date. So in your opinion, what are the advantages of using one pathway over the other? And we can start with any of you, whoever wants to go first. Okay. Rob, I guess you are going first.
So I'd say, again, as Priyanka mentioned, the timelines are pretty similar. So what's the difference between a complex control and a pre-ANDA meeting? So I'll tell you a little bit about the inside process. Is the control correspondences inside our system are generally intended to be, one question for one discipline. The more complex ones, allow consultation with other groups. But the pre-ANDA meeting process is generally a more from the beginning team-based approach to your questions. So the key thing to look, so if you're eligible for both pathways, like it's a complex generic. So some products, a simpler product, might not be eligible for the meeting process, and so you should use the control correspondence process. But if you're eligible for both, like you're talking about a complex generic, what you'll get from the meeting process is a more multidisciplinary team looking at your application, looking at your questions together. And the control correspondence generally will be a little bit narrower, focused on one specific question or a specific aspect of that. And so that would be really my initial idea of, if you're eligible for both processes, why would you pick one? And many of the things that you want to do for complex generics do involve multiple pieces that fit together. Well, I've got to fit together bioequivalence and the device, and I've got to fit together in vitro studies and in vivo studies, and maybe some modeling approaches, and bridge it. There's more things going on in your development program, and the meeting process is intended to give you a more integrated, holistic approach. But if you just have one complicated question on one thing, like, I have a question on, a BE study or a model-based approach, it's very specific and technical, then the control correspondence pathway, could be used there. But if, again, when it's a more range, when the questions range across different aspects of your application, then it's generally better to use the pre-ANDA process than to split it up into several control correspondence, because an integrated approach as well. And not every product is eligible for both of them according to our current user fee agreement.
Like to add to that?
Oh, I was just going to say, Priyanka, I can't hear you, but I was going to start with the concept of whether or not you're eligible. Maybe you don't really have a choice, but I do think that there's also probably an aspect of what are the resources that go into preparing for one versus the other from the industry. I think, for pre-ANDA, there's generally a little bit more of an extensive meeting package, generally a little bit more of a preparation for a subsequent actual meeting with the FDA if it's needed. So, it may be that if you have, like Rob was saying, a very direct question for a single discipline, your resources in preparing for that, submitting that question and getting that response, it may be more feasible or more efficient to do it through control. But certainly, if you're not eligible for the pre-ANDA meeting, then complex control is probably the right way to go.
Yeah, if I can add one more, I think in GDUFA 3, we have now allowed the control correspondence to be asked in other parts of the development program, just not in the pre-NDA space, but also, after a PSG teleconference and even at a post approval or post CRL space and things like that. So So we have generally broadened, sort of the scope of asking those type of questions where, again, you may not be immediately eligible for a meeting, and you already heard that, it uses up a lot of resources for a meeting, both from your side and from our side. But having said that, I think that's important, is that to kind of it's like a menu. Most of the time it's like a menu. You have these meetings and you have the controls and how you want to, and it's not a science, but it's an art, how you want to use that effectively to your benefit. So always think about from that perspective.
Thank you so much. And thank you so much for the engaging discussion. We are unfortunately out of time, so we will hand it back to Corey to talk about all of the questions we have received in the chat. Thank you.
Excellent. Thank you again to our presenters for their excellent presentations and to our experts on the panel for the insightful discussion. We will now begin our question and answer panel with today's presenters. If you haven't had a chance to enter your question into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Our first question is for Dr. Wong. If the RLD is an orphan drug, will the ANDA for the same be acceptable with a batch of 42,000 tablets? Also, what can be included in the controlled correspondence to seek feedback on the same FDA apart from orphan drug designation?
Thank you, Corey, and thank you for the question. Can you hear me okay?
Yes, we can.
Okay. Thank you for the question. I don't think I can give a direct answer in this case in terms of the mentioned batch size, but that could be a valid control correspondence question. In the CC, if you should provide very clear information on your proposal with regard to your batch size, and you should also provide sufficient background information to justify why you propose such batch size for to allow proper understanding and assessment to get the FDA's feedback. Thank you.
Thank you for that response. Another question for you, Dr. Wong. Is the bioequivalence study required to be repeated and submitted to FDA in case of scale up after ANDA approval?
Thank you for that question. I think similar to the previous one, this is a valid question that a prospective applicant can submit post-approval. Without the actual information context, I cannot give you a direct answer in terms of yes or no. So what you need to do is you need to clearly describe your thinking on the studies or bridging studies to support the post-approval scaling up change to see if your proposal would be potentially reasonable or acceptable. So the key here is the clear description on your proposal and related supportive information. Thank you.
Thank you for that. And my final question for you. What is the rationale for not accepting questions on API sameness before excipient sameness has been validated?
Thank you for that question. To clarify, I know I mentioned in my presentation, the one recommendation is you trying to confirm the formulation sameness prior to further steps. That said, the main goal is to make sure you have the formulation confirmed. But that doesn't mean you cannot have another CC if the formulation also requires API sameness. So I think API sameness, if it's required, or formulation Q1, Q2 sameness is required, those are the questions you should try to get further confirmation, if possible, before you move on to more the next step, like the studies that would rely on your test formulation. Thank you.
Thank you. My next question is for Dr. Donnelly. When both PKBE and PDBE rather than one BE study are needed, is CC or ANDA development meetings better suited to get guidance in a relatively short time?
Yeah, thank you for the great question. In this case, you may consider submitting 2 separate CC requests with focus questions on each study. But if the request involves complex issues or multiple questions across different disciplines, a pre-ANDA meeting may be a better pathway. And I think Rob and others discussed this, Dr. Leinberger and others discussed this in the prior Q&A. Thank you.
Excellent, thank you. My next question for you, Dr. Donnelly. Can we file a CC if we need any clarification on requirements stated in a PSG?
Yeah, this is a great question, and the short answer is yes. Clarification of BE recommendations in the PSG would typically be designated as a level 1 CC with a 60-day timeline. Thank you.
Excellent, thank you. My next question is for Dr. Zhang. When submitting a Q1 or Q2 CC, the FDA response only addresses acceptable excipients and does not review or respond at all about the active ingredient type amount, even if the active ingredient is sometimes the main focus of a detailed Q1 or Q2 CC. Why does the FDA not address both the appropriateness of the active ingredient as well as the inactive ingredients for a Q1 or Q2 CC? Thank you.
Thank you for the question. So as stated in the presentation, you are correct. The Q1, Q2 assessment only pertains to the sameness of the inactive ingredient. It does not make sense that generic product have a different API or API amount to the ILD, right? So, but if you have any questions for the active ingredient, for example, the structural characterization, you can always submit another CC for feedback of your active ingredient. We recommend you refer to the control correspondence guidance for additional information when select the appropriate discipline or topic for your control correspondence. Thank you.
Thank you for that response. My next question for you, Dr. Zhang. If proposed ANDA product, which is a parenteral, has been formulated after reverse engineering of RLD, will the applicant still have to seek confirmation from US FDA via CC?
Thank you for the question. The short answer is yes. So in general, we do not ask reverse engineering data of the ILD for Q1, Q2 assessment of parenteral products. The Q1, Q2 assessment is performed based on comparison of the composition table, given the composition table represents the intended target formulation. That is being said, you can still submit a CC for Q1, Q2 assessment in this case. An additional note is that we understand sometimes an applicant may use the drug labeling of ILD as a starting point based on the principle that NDA drug labeling should be consistent with the composition statement for parenteral products. However, due to the manufacturing process of some complex products, variation in certain inactive ingredients amounts may be observed. Therefore, if you believe you received a elective response for your Q1, Q2 assessment inquiry is due to this reason, you may provide a reverse engineering data in another new control correspondence to support your proposed test formulation. Thank you.
Thank you for that response. And my final question for you, Dr. Zhang. If the ANDA to be submitted, thank you for the presentation. If the designated reference listed drug, or RLD, is unavailable in the market, can the sponsor propose alternative approved generic drug products for FDA's consideration in controlled correspondence? Additionally, can the FDA provide guidance via the controlled correspondence on which the approved generic product may be used as the RLD for the purpose of an ANDA submission? What specific information should be included in the controlled correspondence when proposing a generic product as the RLD? Are there any specific FDA guidance documents that outline these requirements?
Thank you for this great question. To answer your question, let us first talk about what is ILD and what is IS, ILD reference listed drug, IS reference standard. The ILD is an FDA-approved drug product that a generic applicant must compare their product to in an ANDA application the ILD serves as a regulatory standard for sameness of active ingredient, dosage form, route of administration, and strength. Also, the Q1, Q2 assessment is performed against the ILD reference listed drug. The reference standard IS is a drug product FDA selected for BE studies in ANDA submission. In most of the cases, the IS is the ILD. They are the same. But if the ILD is discontinued or it is not available, FDA may designate other approved drug product as a IS, and you can check the ILD IS information in Orange Book. If you see the IS, which is a ILD product, in that case, in your development study, it is not available at market. You can send a control correspondence to OJDP asking assignment of IS product due to unavailability of the current IS product. In this CC, you could include the letter from the supplier for non-availability of the current IS product. Also, I recommend that you refer to the guidance for industry referencing approved drug product in ANDA submission for additional information. Thank you.
Excellent. Thank you for that response. My next question is for Dr. Jiang. As part of our CC, we provided a list of ingredients, none of them available as hydration form. Our CC was rejected in part because we did not list the hydration form. How should we indicate the absence of hydration form?
Hello, thank you for the question. You describe it under a specific context of your CC. So just generally from troubleshooting perspective, I would say I can go back, double check against available compendium monographs. I can check with the suppliers for the excipients or other resources to confirm there are no hydration forms available for that inactive ingredient. Or it might be another situation, there are hydration forms, but you are proposing a anhydrous form. Then I would provide my the findings and what you exactly propose in the next CC to help improve it. Thank you.
Thank you for that response. My next question for you. Can Q1, Q2 assessment be used to justify no irritation study or no irritation sensitization study for a transdermal product when Q1, Q2 is not listed as a recommendation in the PSG? Would CC be the right route of communication with FDA in this case?
And thank you for that question too. I think you're interested in waiving the irritation and sensitization study for a product closely matched to the ROD. This is a topic that you can get feedback from the agency through the CC pathway. And as many have mentioned, you can consider the pre-ANDA pathway as well, depending on the overall scope of question you may have. Thank you.
Thank you. My next question is for Dr. Dorsey. If the RLD is discontinued and an RS sample is not available in the market to perform a BE study, please suggest how we can proceed further for performing BE study with respect to RLD RS samples. Thank you.
Thank you for that question. I think Dr. Zhang did a great summation on what is a reference standard. And like she just mentioned, 9 times out of 10, the reference standard is usually the RLD. However, there are cases where it may be discontinued or it's no longer available for procurement on the market. So in those situations, when a CC is submitted to us, we can either provide an alternate approved ANDA for determination of BE. And in situations where, either the RLD is discontinued, the RS
there's no RS, there's no other approved ANDAs, then an applicant can certainly submit a CC for an alternate BE approach. Thank you.
Thank you for that response. My next question for you, Dr. Dorsey. Slide 11 in your presentation recommends that applicants submit separate CCs for varying routes of administration in pediatrics. Can these three be submitted for review in parallel?
So yes, they can. And thank you for this question. So in this situation, even though the CC requests the evaluation of the same inactive ingredient, I'm assuming it's considered a different ask since the CC contains evaluation for that particular inactive ingredient for different routes of administration. Now, this is opposed to a situation where an applicant may come in and they ask for the evaluation of 3 different excipients, the same route of administration, and then immediately turn around and ask another CC for the same 3 excipients, same route of administration, but different amounts. So in this situation of where it is 3 different routes of administration, that would be considered 3 different asks. So they could definitely submit those in parallel. Thank you.
Thank you for that. And my final question for you, Dr. Dorsey. For a new strength to be an oral solid, what is the appropriate correspondence, a PAS or a suitability petition?
Thank you also for that question. This is a good question. In this situation, this depends on the RLD. So if this new strength is not a part of the original strengths within the RLD, then possibly the suitability petition would probably be the best route to go first. However, if the new strength that you are looking to develop is part of the RLD drug product lineup, and you didn't seek to develop that new strength at the original time of submission for your particular application. However, you'd like to do so after getting approval for your ANDA, you'd like to seek this new strength, then of course submitting a control correspondence in regards to a prior approval submission for this new strength would probably be the best way to go for this situation. Thank you.
Excellent. Thank you for that response. My next question is for Dr. Morgan. Would you please repeat the standard weight and body surface area values for adults as you mentioned during your presentation?
Certainly. Thank you for that question. We generally recommend using a standard body weight of 60 kilograms and a standard body surface area of 1.62 meters squared for adults, unless there's something, again, about the particular patient population that suggests that they're outside of a normal range. Thank you.
Thank you for that response. My next question for you, Dr. Morgan. Is the agency working with the international health authorities to have harmonization regarding nitrosamine impurity toxicology assessments? Would information on EMA or others assessment be something to provide in a CC regarding the risks, or would the CC trigger discussion with other health agencies?
Yeah, that's a really good question. There are certainly ongoing discussions to try and harmonize these approaches with other regulators. But importantly, if a product is regulated by the FDA, we would definitely suggest that you use any FDA recommended limits or any risk assessment approaches. And I think it's important just to recognize that we're not going to be discussing commercial confidential information with other regulators without appropriate confidentiality agreements. So, I don't know that necessarily a CC would trigger communication in that way. Thank you.
Excellent, thank you. My final question for you. If the MDD is to be the, quote, highest dose reasonably administered in a day, end quote, if the drug is to be titrated or dosed to effect, what information should be used to develop a proposed MDD value for submission in a CC? What sources of data or justification is acceptable to attach to a CC?
Yes, I mean that is a really good question. Obviously in those instances, that information probably is not going to be available in the dosage and administration instructions and the labeling. We would suggest you could cite different clinical use data from that's published, and you can submit that along with your proposed MDD as a controlled correspondence. That's a really good use case for CCs. Thank you.
Excellent. Thank you for that response. My next question is for Dr. Wang. Can separate CCs for different disciplines be submitted at the same time?
Thank you for the question. Yes, you can submit multiple CCs at the same time as long as each of them do have different questions. Thank you.
Thank you for that response. My next question for you, Dr. Wang. For a generic drug injection with a single formulation or strength concentration that's available at different volumes, I've been asked to send the same question as separate CCs, one per volume. Can you please explain why this is required?
Thank you for the question. Without exact context, it is my assumption you got that request because you were submitting Q1, Q2 CCs. So in general, for Q1, Q2 CCs, because we allow 3 formulations for assessment per formulation, then we recommend generally for like a clean record keeping for each strength, you submit separate CCs for each strength. But if that's not the scenario, then you could, just submit a general question to further clarify with the agency. In general, if your question is not specifically for Q1, Q2, I have not seen personally asking separate CC for each strength. Thank you.
Excellent. Thank you for that response. And my final question for you, is there a length limit for the controlled correspondence, or is a clear question the main requirement as long as it includes the proper justification and rationale?
Yeah, I think, thank you for the question. It is a similar one to the previous. I think as long as each CC has different question with sufficient background information, you could submit multiple CCs at once to the same or different disciplines. Thank you.
Thank you for that response. My next question is for Dr. Donnelly. What happens if the controlled correspondence requester does not respond to the agency's information request, or IR, within the specified timeline in the IR letter?
Yeah, good question. In this case, the agency can close the controlled correspondence request, or in some cases, provide a response based on the limited information provided. Also, it's important to note that the timeline will be extended by the amount of time that the information request is outstanding with the requester. Thank you.
Thank you for that response. My next question for you, Dr. Donnelly. Should the controlled correspondence process be used to seek feedback from the agency when an applicant commenced an in vivo BE study before the PSG was published or revised?
Yeah, that's a great question. Thank you. Here, it's recommended that the applicant requests a PSG teleconference to obtain feedback on the potential impact of the PSG revision or publication on their development program. And then after this meeting, if further feedback or clarification is needed, an applicant can submit a controlled correspondence with specific questions. Thank you.
Thank you for that response. My next question is for Dr. Zhang. If the ANDA to be submitted is for a parental product, will the exact quantity of pH adjuster or buffer to be included composition table while asking for US FDA's feedback on formulation, or only QS will suffice?
Thank you for the question. This is a great question. I also seen several similar questions like this one in the QA box. So if the pH adjuster, the pH adjuster for your formulation is planned to be added based on a QS amount, you should list the amount as a QS. If you plan to use a fixed amount of pH adjuster based on a series of formulation development study, you should list it as a fixed amount. The composition table should be consistent to your manufacturing process. We recommend you refer to the guidance, content, and the format of composition statement and the corresponding statement for detailed information on how to compile a composition table. An additional note is that when the ILD has a specific amount for one particular inactive ingredient, and this inactive ingredient is not an exemption excipient, for example, pH adjuster in this case, then the QS in the test formulation could be considered as Q2 different. That is being said, if you believe you received a negative response in Q1, Q2 assessment inquiry due to the Q2 difference in pH adjuster raised from the fixed amount versus QS, and you believe this difference does not impact safety and efficacy, you can propose a pH adjuster waiver request following our pH adjuster waiver guidance listed in the presentation. Thank you.
Thank you for that response. My next question is for Dr. Jiang. You mentioned that the PSG lists reference standard for comparison in the characterization-based BE approach. Can I compare my test product to the RLD for that approach if the RLD is available?
Sorry, can you repeat your question?
And yes, this question is for Dr. Ying Zhang.
Oh, sorry.
Oh, no, that's fine, and I can repeat the question as well. You mentioned that the PSG lists reference standard for the comparison and the characterization-based BE approach. Can I compare my test product for the RLD for that approach if the RLD is available?
Hi, this is Ying Zhang. Thank you very much for that question. It's a great question. There are situations both the ROD and RS may be available in the market, and we list the reference standard in the PSG page for the characterization-based B approach for consistency. But if you can get hands on the ROD and can get a hold of sufficient amount to support you to use it throughout your characterization-based B approach, yes, you can compare your product to the ROD. Bottom line is, whichever product that you choose, you are expected to use it throughout the studies for your characterization-based B approach as a comparator. And if you anticipate when you submit your CC, there could be confusions about what product I'm going to use, then specify it in the CC submission. That will be helpful. Thank you.
Excellent. Thank you for that response. My next question is for Dr. Dorsey. Can we submit controls to different disciplines at the same time, or do we need to wait for a response to the first one before we submit it to the other discipline? Thank you.
Thank you for that question. And we have had situations in the past where we receive control correspondences that kind of have stacked questions, so to speak. Honestly, maybe one or two offices would have to chime in on this particular case in order to answer it, such as a case of a prior approval supplement. If you're making a change to the formulation or maybe process or manufacturing, we're going off of the recommendations of the SUPAC IR or MR, but at the same time, from an OB standpoint, we may seek the guidance from our OPQ colleagues on the level of change, a particular change may be based off of process or manufacturing. Some of those cases, you know, sometimes we receive them together, and if we need to reach out to another office, then the control correspondence will be bumped to a level 2. However, you know, you can also submit those questions separately, and then they would go to the respective offices to provide their input. So yes, if even though it's related to, say, a prior approval supplement, you can definitely submit those questions separately to the respective office. Thank you.
Thank you for that. I think we have time for one more question, which will be directed to Dr. Morgan. Is review of proposed non-clinical study design or protocol appropriate for controlled correspondence? If so, is there a limit of numbers of studies to be reviewed in a single controlled correspondence?
Yes, thank you for that question. Certainly, a study, a protocol design would fall within the scope of a controlled correspondence, particularly might be useful in cases where the study is complex or a little atypical. I don't know that there's really a limit to the number of study protocols other than to say that you know, it would really need to stay within reason given just the scope of a controlled correspondence. I don't think we would be reviewing more than one or perhaps two. Thank you.
Excellent. Thank you for that response. And that looks like that is all the questions that we have time for. Thank you again to our presenters for the excellent information. I will now turn over the presentation to Dr. Robert Lineberger for closing remarks.
Thank you, Corey. It's been a fantastic event, and I really want to thank the SBI staff and all of our speakers and presenters and panelists and people behind the scenes for putting this together. I think you might have noticed that this is the first event that we've had since the new administration, and so there is a lot of work going on also behind the scenes to make sure this event could happen and get all of that. And, you know, so everyone involved in this really had to go through some extra efforts to make this event happen today. So we really hope that you found it valuable for your work in developing generic products. And from my perspective, I really do want to emphasize the value of this event. You know, OGD receives, and the generic drug program receive 3,000 to 4,000 control correspondence a year. So it's a significant workload. So part of our goal here was to help you use that process more effectively, to ask questions in a way that gets you the answer you need the first time. And, you know, we've tried to be really transparent about that. I know that, you know, from my perspective, there's a lot of little secret knowledge and secret handshakes in the control correspondence process that you have to know. And we've tried to be clear about them here, how to ask the question in the right way that gets you the answer. And hopefully in the presentations, you saw examples of the way to format the questions so that you get the answers that you need. And I think this will create a lot of value. You know, it'll avoid for you you get a like, I can't answer that question. You know, it'll allow us to get quickly to the answer to your question, because we also, from the perspective of the control correspondence program, that if we answer your questions during the control correspondence phase, that'll make your application better. That'll reduce the number of review issues that we have to deal with when we're reviewing that application. So we hope that, you know, you'll use this program effectively, and this will make the whole development system more efficient. And, you know, with that, just to conclude, I hope that if you found this event valuable, you will, you know, let us know, let other people know. You know, the SBIA group has follow-up and surveys and things like that, so where you can evaluate this. So again, if you want these type of events to continue, please share and make sure that it's very visible, the value that these create for your development program, for making affordable generic products available to the American public. And with that, again, thank you all for attending, participating, bringing your questions to this. And I want to turn it back over to our SBIA colleagues, and they'll finish up the final housekeeping and results on that. But again, from my perspective, thank you all very much for participating in this event.